Hello, and welcome to our presentation on forensic DNA quantitation, which is the third step in the DNA workflow. So we've extracted our DNA, and that's great, but we still have a few questions we need answered. First of all, we don't know how much DNA we actually have present in our sample, and of that DNA, we don't know how much of it comes from humans, and humans in a forensic setting are what we're interested in most of the time. So we do what's known as DNA quantitation, which is just calculating how much DNA we've got in a sample. And ideally, we also want a process that's human-specific so that it only calculates how much human DNA is in that sample. This is also required for PCR, which we'll get into later, but is a necessary step in the DNA process. So before going into the details of quantitation, we just want to review some additional reasons as to why we want to perform this. First of all, the FBI requires it. Quality Assurance Standard, or QAS 9.4, says that crime labs must perform quantitation. Thou shalt quant. Additionally, we need to do quantitation for better PCR and STR results. Exactly what these acronyms mean we'll get into later, but in short, it tells us how much DNA we need to take for upcoming reactions. If we add too much DNA, then it can overwhelm the reaction and the instrument and we'll get poor results, and too little DNA will lead us to not getting any results at all. Then quantitation levels that we get back may also give us additional information about things we should or should not test with the sample. So for instance, if our quantitation levels are really low, then we can try using some low copy number techniques to give us more DNA to work with. And then finally, it can help us narrow down potential sources of error. So if we get poor quantitation results, maybe we need to re-extract the sample, and then later on in our process, if there are problems that occur, we can know that the DNA template or a quantitation was not the source of the error and it was something else along the way. So we've brought up PCR a few times, and it stands for polymerase chain reaction. Though another group is going to go into more details about this later, we do need to touch on it here because it's important for the quantitation process. The polymerase chain reaction duplicates or amplifies a particular segment of DNA, and there are three stages that comprise a thermal cycle, or a cycle of heating and cooling. These are denaturation, annealing, and extension. By the end of a cycle, the important thing is that we have more copies of that particular segment of DNA. Now, for PCR to occur, we also have to make sure that we add the necessary components. These include the DNA that we're wanting to duplicate, primers, DNTPs, like adenine, thymine, cytosine, and guanine, and Taq DNA polymerase. We then repeat the thermal cycles around 40 times, and after the first few cycles, the DNA quantity doubles. The technique we like to use for PCR is known as quantitative real-time PCR, or qPCR. Here you can see a photo of the instrument we use for that, which is the 7500 real-time PCR system, and it measures the fluorescence of the DNA in real time, or once every thermal cycle. This is in contrast to, say, measuring the fluorescence at the end of all thermal cycles. The fluorescence comes from fluorescent dyes that we attach to the DNA. Some examples of these dyes include Cyber Green and the TaqMan probe. We're going to discuss the TaqMan probe because that's usually what we use in our labs. There is a direct correlation between the amount of fluorescence in a sample and the amount of DNA in a sample, which makes sense because more DNA means more of the fluorescent probes can attach to the strands and thus fluoresce. Now, fortunately, we don't have to mix everything needed for the qPCR reactions ourselves. There are some companies that make kits that we can use that have been verified and tested so that we know they are reliable. The three main companies in the United States that manufacture these kits are QIAGEN, Promega, and Thermo Fisher. The kit featured in this picture is the QIAGEN Investigator Quantiplex Pro Kit, which is the one that we often use.
There are four general stages to the qPCR process. During the initial baseline stage, the PCR product is not above the baseline noise. During the exponential phase, the PCR products are roughly doubling. This is also the stage where we use fluorescent dyes to measure the amount of DNA created in the qPCR reaction. During the linear stage, the amount of PCR products is increasing at a steady rate. Lastly, the plateau stage is when the amount of PCR products is no longer increasing due to the exhaustion of the PCR components such as primers or DNTPs. After extraction, the DNA extracts are loaded into the sample plate along with the reagents necessary for qPCR. These reagents are provided by the commercial qPCR kit being used. Standards, also provided by the kits, are loaded into the first few wells to provide a baseline comparison that is used to determine the amount of DNA present in the samples. Once the samples are loaded into the sample plate, they can be placed into the qPCR machine. Old wells are removed from the qPCR, and the new samples are placed into the tray holder. The qPCR machines are pre-programmed with the specifications and cycles needed for each commercial qPCR kit. Once samples are placed in the machine, a program for the kit being used is chosen and qPCR is started. This process can take between 30 to 60 minutes depending on the number of cycles. The TaqMan probe is a standard fluorescent dye used in forensic DNA testing. The probe is designed to bind to a portion of the targeted DNA strand for replication and consists of a reporter dye and a quencher. During qPCR, the DNA polymerase molecule binds to the forward primer and begins extending the primer until it encounters the probe. When the DNA polymerase molecule encounters the TaqMan probe, it will digest the probe, releasing the quencher and reporter dye. Once the reporter dye is not connected to the quencher, it will begin fluorescing. The amount of fluorescence of the reporter dye is used to determine the amount of PCR products in the reaction, as the number of reporter dyes fluorescing represents the number of probes digested by the DNA polymerase and thereby the number of PCR products created.
The cycle threshold is a threshold used to determine if a qPCR is successful. If the amplification curve does not pass the threshold, then there was not enough template DNA in the reaction. In PCR, typically the amount of template DNA or PCR products doubles each cycle. In general, unless there is a lot of inhibition, a higher amount of DNA will result in a lower cycle threshold value, and less DNA will result in a higher cycle threshold value. The cycle threshold values and amplification curves of known standards are used to determine the amount of DNA in the questioned or crime scene DNA sample. Standards are pristine, synthetic, and undegraded DNA samples that should result in optimal PCR results. These standards represent known amounts of template DNA. By comparing the amplification curves to the questioned DNA sample, based on which curve it falls between, it is possible to determine the amount of DNA present in the DNA extract. As a result, your quantitation results are only as good as the standards that you use, so quality control checks of standards are important to ensure accurate qPCR results. These standard curves are examples of the degradation target and human target. Generating standard curves helps us determine if a PCR was efficient and free of contamination. As mentioned, the results of a qPCR are only as good as the standards used in the reaction. This is an example of what a quantitation report looks like. We'll focus briefly on the degradation and human targets. The degradation and human target ratios will indicate whether a sample is degraded or not. The human target specifically is the quantitation used for normalization. For normalization, quantitation determines the concentration of DNA. In STR typing, there's a specific amount of DNA required, 0.5 nanograms of DNA. Normalization is the process of determining the volume that contains the target DNA amount. Dilution formulas or ratios are used for calculations to determine the volume that allows us to obtain 0.5 nanograms of DNA for STR typing. And that is based on concentrations obtained from quantitation.